Good evening. My name is Hafsa Rabbas and welcome to Season 2 of Understanding Cancer. Tonight is Lecture 1 and it's titled Introduction to Cancer. So what will we be learning tonight? What is cancer? Can we actually cure it? The importance of cell communication, a brief insight into the history of cancer, an overview of the DNA, genes and chromosomes, and to find out what causes our genes to become faulty. An ideal way of learning is to divide these lectures across the 7 days. The approximate total time for this lecture is 1 hour. And you can do this at your own pace, and this can then be followed by challenging yourself with a quiz. So how can you support your learning? You can support your learning through the key facts with diagrams that are designed by HN Designs, and this is presented in a simplified way that is suitable not only for students but also the general audience. There is a glossary to help you understand what keywords mean. There are summary doodle revision posters to help consolidate your understanding. There are also quizzes to test your knowledge and reflect. And there is a reference list for further reading for anybody who is interested. A special thanks to my parents, family, friends and colleagues for their support, and the respected teachers and health professions who taught me and installed the passion of cancer. So let's begin with what is cancer? Can we actually cure it? Cancer is a disease characterized by uncontrolled growth of the body cells. Now, if we look at the first image presents an initial tumor cell that has a change within the gene, and this is known as mutation. So when there's an initial tumor cell that has a mutation there, this is known as benign growth. Now, this can continuously grow, and this is known as malignant growth. And it can invade and metastasize to other parts of the body. In other words, it can spread from the original sites where there's the benign growth and a primary tumor, and it can spread to other parts of the body to form secondary tumors that are malignant and invasive. So how does it travel to other parts of the body? This is achieved through the blood and also lymph vessels. Lymph vessels are tube-like structures that carry fluid and white blood cells to fight infection and disease. Just as a recap, please do visit Season 1, where we discussed what is a cell, what is a cancer cell, how you can tell the difference between the two, also the causes of cancer and how cancer can be diagnosed. Cancer is the second most leading cause for deaths around the world, and it accounts for 9.6 million deaths in 2018. Cancer Research UK reported that between 2017 to 2019, 167,142 deaths were caused by cancer alone. And it was reported by World Health Organization that in 2018, half of all of the cancer deaths were associated with the lungs, the bowel, the breast, and also the prostate cancer. Now the question here is, after discussing the mortality rates, is there a cure for cancer? There's always hope, so there's a possible yes. So who knows? The number of cancers can be treated successfully, but it depends on the following factors: age, when was it caught, and the lifestyle factors. For example, is the patient elderly? How strong is the immune system to fight off infection? When was the cancer actually being caught? Was it caught early or at a later stage? Or was it actually being treated and returned again? Lifestyle factors, for example, how much exercise does the patient actually does? What do they eat? Do they actually smoke? How much alcohol do they take? And the area of work, for example, do they utilize a lot of chemicals? Is there a lot of gas being released? Do they work in mining? Is there any factory work being present? However, with the improving methods and advanced technology that is being made over time, cancers can be detected early and hopefully be treated before they can become life-threatening. However, if it is caught at a later stage, the primary goal of treatment for metastatic cancer is to control the growth of the cancer or to relieve the symptoms it's causing, because this can have a huge effect on how the body works. So cancer education is not only just important for patients and their families, but also for the community as a whole and health authorities. So collectively, it can lower the burden. Let us now proceed on to cell communication. So why is it important to understand why or how cells communicate? Because cells can sense and respond to signals in the extracellular environment. Now, if we look at the image over here, you can see that there's the plasma and you also have the interstitial fluid. So interstitial fluid is the fluid between the cells. So the extracellular environment is the area outside the cell, and it consists of two parts, the plasma and the interstitial fluid. So extra means out and cellular means the cell. So the role of the plasma membrane is to separate out the intracellular from the extracellular. So intra means in and cellular is the actual cell. It's important also to understand the cell communication because cells also help coordinate and regulate activities within the whole organism, whether that's a plant, whether that's an animal, whether that's a human. In addition, the cells communicate to one another, especially adjacent cells. So if you look at this diagram over here, you can see that there's two cells that are joined together and they don't really have any cells between them. So this is known as adjacent cells. So this can also affect the behavior of cells that are close and distanced. So what is a signal? A signal is a substance that influences the properties of the cells. That could be, for example, a hormone, a protein. And amongst these cellular activities is growth and how cells divide. So let's discuss a brief insight into the history of cancer. Edwin Smith. So there are a number of scientists that help understand what cancer is today, whether it's from a cellular basis, whether it's through the molecular or gene basis, whether it's through an enzyme level, or whether it's through the history and discovering these aspects. Everybody actually played a role, okay, or developing equipment to help understand and this. All of these factors help shape our understanding of what cancer is today. And it's unfortunate that we can't discuss every single scientist or every single archaeologist that partake on this journey. However, we'll provide a brief insight to a few of them. So let's begin with Edwin Smith. So Edwin Smith, he visited Luxor in 1862, and he discovered the earliest record of cancer that was done on Egyptian papyrus, and it was estimated that it was 1600 BCE. So here is an image from the National Library of Medicine in 2016, and you can see that there are records of cancer patients on the Egyptian papyrus, which was discovered by Edwin Smith. So how was one be able to understand the hieroglyphics that are present on the papyrus? Please meet James Breasted. So in 1925, he translated these records at the University of Chicago, and at that time within ancient Egypt, there were 48 cancer cases recorded. And one of the common symptoms that has been established amongst the patients is abscess. So abscess is an infected area that contains pus, and pus is dead cells and bacteria. And it was treated using cauterization. And cauterization is a procedure where there's a burn to the skin or a wound, and using a hot device. So in this case, it was a red hot iron. And there were also records of patients that had swollen or bulging tumours. But the physician, who was unknown, didn't make any records in terms of the treatments, which would have been interesting to find out. And in 2018, 6 bodies of cancer patients were discovered in the western desert of Egypt. Please meet Hippocrates. So in 400 BCE, Hippocrates discovered that tumours were rich in blood and also resembled the limits of the crab. Okay, so he also discovered that gangrene, so there's tissues, there was blockage or there's decay, and this is because of infection or limited blood supply. And there are several number of keywords that have been derived from the Greek language, which have been anglicized and used today. For example, we have onkos. So within the English language, this means swelling, and the medical profession for cancer is oncology. So the oncology, onkos, so that's how the link between them came. In addition, you also have carcinos. So carcinos within in English is actually carcinoma, and it's the most common type of cancer that actually begins with the skin and the lining of the actual organ tissues. And apoptosis is a Greek word, and it's within English it's like a programmed cell death, and it means dropping off. 600 years later, Galen made some records on cancer patients he treated, and he also defined cancer. So the Latin word for crab is cancer, and that's what is being utilized today. Following this, please meet Ibn Sina or Avicenna. He's a Persian physician who wrote Canon of Medicine in 1025. And his book predominantly was used for study, especially in medical programs, and was considered as an encyclopedia. And it has been translated in a number of languages, and amongst them is Latin. He was also considered as the first cancer surgeon, and he utilized plants as another treatment strategy for such cases. Joseph Needham, he studied Chinese medicine a lot and also worked in farming and agriculture. And through this method, he has developed a number of ways to help cancer patients. Let us now proceed on to an overview of the DNA, genes, and chromosomes. Now, our body is made up of millions of cells, and inside the cells there are little organs called organelles. So in this diagram over here, you can see the basic image of what an animal cell looks like. So you have organelle called mitochondria. So this is helps produce energy. The ribosome, this helps produce protein, which is needed for growth and repair. And the cell membrane, this helps controls what enters and leaves the cell. And you also have the nucleus. The nucleus is key into controlling the cell and its activities. So how does it do this? It's because it has genetic information in the form of the chromosome here shown. So what are chromosomes? Chromosomes are long thread-like structures made of DNA. Now if we look at this diagram over here, each part of the chromosome is known as a chromatid. Okay, and when you have two of chromatids joined together, that's known as sister chromatids. The center of the chromosome is known as the centromere, and the ending of the chromosomes are known as telomere. The chromosomes have a short arm, and that is P, and it also has a long arm known as Q. In every body cell, there are 23 pairs of chromosomes. For example, if it's a heart cell, kidney cell, muscle cell, they all have 23 pairs of chromosomes. However, this excludes cells that are associated with the reproduction. So we're referring to the sperm cell and the egg cell. So the sperm is found in the male reproductive system, and then you have the egg cell that is found within the female reproductive system. And they have 23 chromosomes only. Here in this image, you can find a diagram that presents a more detail of what the animal cell consists of. And here there's also the plant cell and the bacterium. So all organisms are made of cells. So far we discussed the nucleus, the chromosomes, and now we will discuss the DNA. DNA is an abbreviation for deoxyribonucleic acid. And this is a two-stranded DNA that are coiled together or twisted to form a double helix. The diameter of the DNA is 28 or 2 nanometers. The distance between the two nucleotides is 3.4 A, and the length is 34 A. It is long, it is also a thin, and the two DNA strands consist of short subunits or monomers called nucleotides. They are presented in an anti-parallel procedure. So if you look at the diagram, one of the strands begins with the 5 ends, and the other strand begins with the 3 end. So in other words, they are anti-parallel, and they're twisted to form a ladder structure. So who helped us understand the structure of the DNA? So please meet the Watson-Crick model of the DNA. So before Watson and Crick, there were other scientists, Frederick Miescher, P.A. Levine, W.T. Asbury, Maurice Wilkins, and Rosalind Franklin. And they helped Watson and Crick to understand the structure of the DNA structure. How? They utilized a process called X-ray crystallography. And this helped discover the image of the DNA through a procedure called diffraction. And this helps Watson and Crick to look at how the DNA looks like, the structure, what it consists of, And this is what led them to develop the model in 1953. So here is an insight of the structure of the DNA nucleotide. So a gentle reminder that the DNA consists of 2 strands that have many short subunits called nucleotides. And each nucleotide is divided into 3. Phosphate group, so this is a phosphorus element that is attached to 4 oxygen atoms. You have the sugar molecule. The sugar molecule, as you can see, it has like a pentagon shape. So it's a 5-carbon ring structure known as a ribose. There is no oxygen group in the food carbon, which is why it's called deoxyribose rather than ribose. And the base has lots of nitrogen, as you can see within the image there. So it's a nitrogenous base. And there are 4 types of bases. There's adenine, cytosine, guanine, and thymine. And this means there are 4 types of DNA nucleotides that can be formed. So what that means is that the sugar and phosphate remains the same, but the base, because it's 4 different types, that means there's 4 different types of nucleotides. And the bases are joined by hydrogen bonds. And they do not have a random process, but they have a complementary base process, whereby there is a specific base joins with another specific base. So adenine always joins with thymine, and guanine always joins with cytosine. So adenine joins with thymine with 2 hydrogen bonds, as you can see within the image there, and the guanine always joins with cytosine with 3 hydrogen bonds. A step further, the bases can be divided into purines and pyrimidines. Purines are 5-membered ring structures, and they are adenine and guanine. The pyrimidine are double-ring, 6-membered ring bases, and they consist of cytosine and thymine. So a purine always complementary binds with a pyrimidine via hydrogen bonds. So a scientist called Chargaff, he developed certain rules and principles to help understand the sum of purines and pyrimidines. Now, the sum of the purines always equals the sum of the pyrimidines. So what that means is that the adenine and guanine base composition equals the base composition of thymine and cytosine. However, this cannot be achieved by the complementary base pairing. So the A and T, despite they join together, they do not equal in terms of the base composition to guanine and cytosine. It's only if the purines are joined together and the pyrimidines are joined together, that's when there's an equal number. So the sugar-phosphate bond is what attaches the nucleotide to another, and it's called a phosphodiester bond. And it's negatively charged, and it's what helps the DNA to become water-loving and form bonds with water. Okay. So the phosphate group is attached to the 5th carbon of the sugar on 1 nucleotide, as you can see within the image there. And the 3rd carbon of the nucleotide is where the 3 hydroxyl group, i.e. OH, is attached to. OH consists of the oxygen and hydrogen. Okay. The base is attached to the 1st carbon. So all this can be seen with this image over here. So these bonds are what form the 3 to 5 phosphodiester bonds, and this is what provides stability for the DNA. So far we discussed the structure of the DNA. So let's look at the structure with the function. So what are the function or the role of the DNA? It's storage. It stores all the genetic information to help build an organism or life form. How it can store information is because the length helps it to be compacted in such a way that it can provide a small volume, but it can be accessible. It also helps stores instructions necessary to make proteins needed for growth and repair. And it also helps to provide an energy store for facilitating the transit of enzymes called DNA and RNA polymerases. So what's the origin of the DNA? Well, 50% comes from our mothers and 50% comes from our fathers. Okay, so there are 2 copies for every gene. What's a gene? A gene is a short section of DNA. It contains instructions to help or tell the cell what to do. It's also estimated that there are approximately 25,000 genes in humans, and this can affect and control how we look like, in other words, the phenotype, how we behave, and when to grow and divide. Proteins are produced in the ribosomes, so these could be cut in small sections like fragmented, could be multispecific, or there could be whole proteins. So let's look at the image of the animal cell there. So here's the ribosomes. So this is where proteins are being made. So what's the function or the roles of the proteins? They help speed up chemical reactions as enzymes. They help regulate body functions as hormones. They help protect the body from infections via immunoglobulins. And they also provide structural supports and muscle movement. Genes have the information on instructions needed to make these proteins. So it consists of 2 processes, transcription and translation. So transcription is where the DNA is being used as a template to make the RNA molecule. The differences between DNA and RNA. DNA is a 2-stranded molecule, whereas the RNA is 1-stranded molecule. There's also that the DNA has a deoxyribose sugar, so there's a removal of the oxygen, but the RNA keeps its oxygen, so it's a ribose. There's also differ in terms of the bases too. Okay, so the DNA is used as a template to make the RNA molecule, and in terms of translation, this is where the RNA leaves the nucleus and it travels to the ribosomes to translate the information, okay, that is provided as a template to make the protein. So what causes our genes to become faulty? The root cause of cancer are mutations, and mutations are random changes in the genes. And this is what helps the cancer cells to grow quickly, and this is known as hyperproliferation. And it allows the mutations to that even affect how normal cells divide and grow. So there's unstable growth, and this can not only what causes cancer, but also where the disease is. So if you look at this image over here, you've got a normal cell, then that's proceeds onto a first mutation, second mutation, so you can see the mutations growing. Then you've got the 3rd mutation, 4th mutation, and then when it reaches the 4th mutation, you can see that it's converted into malignant cells. So that's how it invades and metastasizes. So what are the different types of mutations? So mutations, there's different examples. So you can have copies. So when you have multiple copies, that's amplification. You can have a removal or deletion of one of the base pairs. You can have an addition or insertion that occurs within the sequence. You can have chromosomal mutations, and you can have inversions. So there's a variety of types of mutations. And what increases the risk factors of these mutations is spontaneous mutation. So this can be a natural process from our parents, or it can be associated with the environment, so it induced mutations. So that can be associated with the chemicals that we are exposed to. So if it's chemicals released in the air, if it's tobacco smoke, if it's radiation or radiation from the sun, or if you're having a procedure such as X-ray and gamma rays. So key facts. So the first type of gene is proto-oncogene. So it has a stimulatory effect. So it has like a encouraging effect. So they are normal genes, but when they become mutated, they become oncogenes. So these oncogenes, what they do, they allow the cancer to actually grow. And there are about 70 proto-oncogenes in our DNA. And the key example is BCR-ABL protein in blood-related cancers such as chronic myelogenous leukemia and acute lymphoblastic leukemia. So about 95% of CML cases and some ALL cases. An oncogene. So earlier we mentioned that an oncogene is a proto-oncogene that has been mutated, and therefore there's an overactivity there. There's unregulation in terms of the growth, and it's not really actually passed from the parents. And there's key examples over here is HER2. So this is what's found in 30% of the human breast cancers. You have the RAS gene that controls the transcription of the genes, regulating the cell growth and differentiation. You also have the mice protein, which is a transcription factor, and it controls the expression of several genes. And you also have the SRC. So this was the very first oncogene that's ever been discovered, and it's a type of enzyme called tyrosine kinase, which helps regulate the cell activity. And you have the hTERT, and this is code for an enzyme called telomerase. So earlier we discussed the chromosomes, and we mentioned that the ending of the chromosomes is known as a telomere. So the telomerase has a special role to maintain the chromosome ends, and we will discuss this when we look at the hallmarks of cancer next lesson. Tumour suppressor gene. So as the name suggests, it suppresses the tumour. It prevents the cancer from occurring. So it encodes proteins that does that. Okay. How it can be achieved is through either slow or stopped cancer growth by DNA repair, self-destruct via a process called apoptosis, so which is programmed cell death. Now, when you have just one copy that's been mutated of the gene, it doesn't actually cause cancer, but it actually increases the risk of cancer because you still have the other copy that's normal and functional. Okay, because remember there is 2 copies for every gene, 50% from the mother, 50% from the father. When you have both copies of the gene that are malfunctions, there's a loss of function there. So that's what gets mutated and causes cancer. So if you look at this image over here, you have the oncogene. So over here we've got when one affected, it can result in abnormal growth without any form of regulation. Okay, when you have a tumour suppressor gene that are both are affected, there's a loss of growth inhibition, and that can lead to abnormal growth. And when you have just one affected, it's haploinsufficient, so it can still cause a lower gene dose, and that can possibly increase the risk and lead to abnormal growth. So what are the key examples of tumour suppressor genes? You have P53. P53 is a transcription factor that regulates cell division and cell death. You have P14ARF that prevents the degradation or the destruction of P53. You have retinoblastoma. This includes for the retinoblastoma protein, so it can affect or change the activity of the transcription factors, and this is how it can control cell division. APC, adenomatous polyposis coli. So this regulates tumour growth, how cells stick together, so adhesion, how cells move, so migration. And when you have a removal or inactivation, as in, for instance, it's not working, then the APC is linked with colorectal cancer. And you have VHL, which is von Hippel-Lindau, and that leads to blood vessel tumours and kidney cancers. So key examples are several hemangioblastoma and renal cell carcinomas. You have CDKN2A. So this is what encodes to p16 Inc. for a protein that functions in how cells divide, differentiate, and die via senescence and apoptosis. You also have the breast cancer gene. So this regulates the DNA repair and cell cycle. And you have the p10, which is phosphatase and tensin homolog. So this regulates cellular activities by inhibiting how cells move, how cells grow and survive, by inactivating a signaling pathway, which is by an enzyme called PI3 kinase dependent. You also have WT, so that stands for Wilms tumor. So WT1, WT2, they are transcription factors needed for normal cellular development and survival, and WT1 plays both an oncogenic role as well as suppressing tumors. And NF1, NF2, neurofibromin, that negatively regulates the RAS oncogene. So DNA repair genes. So far we looked at proto-oncogenes, then oncogenes, and now tumor suppressor genes, and now we've got the DNA repair genes. So the DNA repair genes, they code for proteins that correct the errors. So this helps before cell division takes place. So, but the problem is when there's mutated versions of the DNA repair genes, it's like a domino effect, so it can affect other genes such as tumor suppressors and proto-oncogenes, leading them to genetic mutations there. And it can also change the chromosomes. So then when there's a removal or when there's an additional, so like a duplication, and this is what can cause a cell to become cancerous. Key examples of this is BRCA1, BRCA2. These are examples of DNA repair genes. And you have ataxia telangiectasia mutated ATM. So that's released when there is cellular damage or that plays a role in cell cycle progression. So we've reached the end of today's lecture. I hope you enjoyed it because I did. And we're just going to go through key points. So cancer is a disease caused by random changes in the genes called mutations. This leads to uncontrolled cellular growth and the primary site that can spread to other parts of the body to form secondary tumours that are malignant. A number of scientists and archaeologists have helped shape our understanding of cancer. Cell communication is important because it helps us understand how cells sense, how they respond to signals, and how they coordinate activities. From the largest to the smallest size, we'll begin with a cell, nucleus, chromosome, DNA, and a gene. Genes, they encode the proteins. They have the information, they have the instructions needed to make the proteins. And this requires a two-step process, transcription and translation. Genes can become faulty. How? Through mutations, and these mutations can be either passed from parents, so spontaneously, or can be environment which are induced, so it's like the chemicals involved. So the proto-oncogenes, they stimulate cell division. The tumor suppressor genes, they inhibit cell division. The DNA repair genes repair errors that occurs during cell division within the cell cycle. So what's next? So we went through the lecture. You can have a read, make some notes, use the glossary to make note of what you learned and highlight any points. Reinforce your understanding. Why don't you try a quiz? It's a fun way of learning by yourself or with a friend. Check out the summary posters. It summarizes key concepts of this lecture. If you're interested to learn and read more, have a read on those further reading lists. Plenty of nice resources there. If you require any further assistance, I'm more than happy to have a 10 to 15-minute meeting on Zoom to discuss further in terms of anything you misunderstand within this lecture. So here is the reference list for anybody who's interested for further reading. Next week, we'll be discussing the hallmarks of cancer, so what characterizes the cancer. So I hope you have benefited from this lecture. Take care. See you next week.